All right, good morning, everyone. I hope you had a good last 2 days. Your quarter is going smoothly. I'm just going to give people 30 seconds to roll in, and then we are going to get started here. Just a note that what I'm asking you to do today is a little bit different in your sign-in. Please do first put in your last name, first name. So last name comes first. That makes it easier for me to track that you were here when I'm assigning points for participation. But I also would like you to add your lab group, so it'll be easier for me to put the breakout rooms. If you're not sure which lab group you're in, I assigned them this morning to anyone who wasn't done, or if you don't remember, you can find it on Canvas in the people under the lab groups tab. So we'll just take another minute to let people do that little sign in. All right. Oops. Okay, so just a reminder as you're coming in that I'm asking you to sign in with your last name, first name, and lab group. I see most of you are remembering to do that, but just a reminder to please sign in with your last name, first name, and lab group, please. Thank you. If you've already put in your information but didn't do the lab group part, put that in as well. All right, so it's time to get started. The video lectures, so let me take a step back and just say what I am expecting you to have already done by today. Since we're starting a topic in chapter 1, the sort of introduction to microbiology as a field, before every topic in the Zoom section, I expect you to have actually gone to the topic section and read through all of the slides, watched the video lectures, read the textbook if you have it. I know some of you don't have it yet, that's fine. The textbook is sort of secondary in my opinion. I think the most important thing is to watch that video lecture. So you already have been exposed to the material, take the notes that you would normally take. So you're kind of ready. You understand the material at least at first blush. And also there's usually a pre-quiz, although not this week because you had a lot of other things to get organized on, like the lab groups and orientation quiz and stuff like that. But normally there'd be a pre-quiz as there is next week for microscopy. So I'd expect you to have done all that prep and done the quiz and then show up here kind of with your mind ready. And a clarification or just a reminder that the Zoom sessions are not comprehensive. You're responsible for more than what we talk about here. What your primary study source should be is the video lectures that are on Canvas, which you can rewatch and pause as much as you need to. So in this Zoom session, what we do is try to bring the material to life. We try to address some of the concepts that are a little trickier to just learn on your own and really try to engage with the material, but I can't get to everything in just 2 hours. Okay, so the major questions addressed in this topic in chapter 1, Introduction to Microbiology, are basically what are the microbes that we're studying? So how do you define them? What is a sort of a notable feature about them that's a definitional feature? And then we also address this question, which is a little bit more loosey-goosey. Why do microbes matter? What are their different relationships to us? There's a history section. So how do we know about microbes? It's a field, as we talked about last time, that's really been driven a lot by the technology that allows us to see into this tiny world and to understand it better. And by see, I don't just mean see with a microscope. Sometimes the way we see into this world is with sort of indirect biochemical assays. And then we'll talk, in this section, we address the current challenges, sort of what is modern microbiology doing And it's very vast, this topic, so we barely scratched the surface of it, honestly. So let's start with just a definition of the word microbe, right? It just means an agent, an organism, which is like a living cell or in some cases a multi-celled organism, or some non-living infectious agents that are too small to be seen with the naked eye, but they are still sort of biological in a way. So majority of microbes that we consider are actually cellular life forms, most of them single-celled, some of them multi-celled, that are very small. We can't see them with our own eyes. We need a microscope in order to see them. And we also consider some infectious agents that are not technically alive in the same way that cellular life is alive, but they're still infectious, they can replicate, and so we consider them in this section as well. There's obviously a lot of things in the universe that are smaller than the naked eye that we don't consider in microbiology because they're not biological. Okay, so this is just a link that you can go to on your own to kind of get a sense of scale. And I'm emphasizing it here only because when you go there, you might feel like this is cute and fun, but a little bit silly. But in my experience, one of the challenges of learning this field, unlike say anatomy and physiology, which you just completed, that has its own challenges. There's a lot of, heavy material, a lot of stuff to know. But it's easier for us to visualize and remember that stuff because it's about our bodies, which we live in all the time. And we can see a lot of those parts. Like if someone says, where is your elbow, you can actually point to it. You've looked at it your whole life. But when you think about cell biology and microbiology, it's a little bit more abstract. We have to have a picture in our minds and we have to get familiar and friendly with this world. And so it can be quite helpful to use these simple tools to kind of just ground us in understanding what we're really talking about and some of the relative scales that we're thinking about. So I highly recommend that you do this. Just to give you a sense of the scale of things, things that are prokaryotic, that is those single-celled eukaryotes that are obviously mostly microbes because we can't see them. One of the largest ones would be an amoeba. Now this one is so big that actually you could see it with your eye probably as just a really tiny dot. And some of you looked at amoebas under a microscope in cell biology and maybe even noticed that when you were looking at the container they were in, you could see these floating specks. So we'll talk about next week the sort of where's the limit of what's big enough for us to see. But this is an example of something that is technically microbial, but it is sometimes large enough that we see it as a tiny dot. So this is a eukaryotic single-celled organism that's considered a protozoan because it's animal-like. We have then something that you might be familiar with, a human red blood cell, which is one of the smallest human cells. It's 10 micrometers in diameter. And yeast, which is a single-celled fungus, it's another eukaryote, is a similar size. 10 times smaller than that is bacteria, like E. coli. And this is actually not its true size because it's a rectangle or a rod shape, so it's skinnier than it is long. But this is about the scale. It's around the 1 micrometer scale. And then down here we have flu and coronavirus, very similar in size, which is 10 times smaller than that. So we've known about microbes for a lot less time than they've been around. They've existed for billions of years, long before we showed up. We learned of their existence about 4 centuries ago because of light microscopes, but actually only really started thinking about them as important for the last 200 years. And so now we're in a world where we realize that microbes are crucial to many aspects of our own lives, to industry, because some of them do industrial or metabolic processes that we can't do, ecology, because out there in the world, lots of microbes are doing all sorts of things that you don't necessarily think about, but are actually critical to our own survival. Obviously, very famous are the subset, very small subset of microbes that are pathogenic and hurt and kill people every year. All right, so in the chapter 1 video lectures, you will have learned about all of these different figures in history. And so I'm going to give you, again, this is not a comprehensive, but this is just to see, like, do we remember some of these guys and what they do? We're going to do a little matching exercise. So I'm going to say a contribution, and then in the chat, you're going to write the name of the person who did that. So who was the first person to name cells? Great. Yes, that was Robert Hooke. So here are your options up here, just in case and I would expect you to know these cold, but I have provided these up here to sort of refresh your memory. Who pioneered the use of antiseptic technique? I find this one easy to remember because Listerine, right? Listerine, some of you may be old enough to know what that is. That was Joseph Lister. Who discovered the first antibiotic famously? Gloria, go ahead, your hand went up.
Sorry, that was accidentally.
Oh, that's absolutely fine. Thank you. All right, good job, you guys. That was Alexander Fleming in 1928, who provided strong evidence for biogenesis theory, in particular, as it relates to microbial life. Strong evidence for biogenesis as it relates to microbial life. Other people had done pretty good experiments that convinced people that macroscopic life, like us, is biogenetic, but there were skeptics, right? Yes, that was Louis Pasteur. He did lots of things. That was one of his many contributions. Who pioneered vaccinations, at least according to the historical record. There's, hundreds of years of people doing versions of this, actually back to ancient China. Yes, that was Edward Jenner. Good work. And who was the first to observe living cells? This is a little bit tricky because actually, if you know history too well, then that will be hard to answer this question, but who is traditionally credited with this? Yes, that's right. Yes, that was Antony van Leeuwenhoek. Good work. Okay, now we're going to do a more difficult one. So I have paired scientists in these groups because they have made a contribution that sort of relates to the other person. So they've done something together, or not together, not working together, but rather their contributions are related in terms of what they contributed to the field. So I'm going to cold call people for this. All right, so be ready. And also don't forget that it's okay to be wrong. I will not hold that against you. I just ask you to try. Or if you're feeling really unsure and don't want to be on the spot, it's also always okay to pass. And there's no shame in that at all. And when you do that, if that's what you want to do, you give other people permission to do that as well. So I appreciate you. Okay, so with no further ado, Sabrina says, we don't need to know the dates. You need to know rough centuries, okay? So what group did what and to the nearest century? So was it 1600, 1700, 1800, 1900? You do need to kind of know roughly the order these things happened. And I don't care if it's like you're off by, 10 years, but it's best to know the century. Thanks for asking. Okay, here we go. So which group debated the origins of life, biogenesis versus spontaneous generation? I'm going to cold call somebody. So I just want to give you a second to think about this. All right, and let's go to Chandler. What do you think?
I think that, let's see. Is it too quiet?
No, I can hear you.
It should be working.
I can hear you. Can you not hear me?
Oh, you can't hear me? Okay.
I can hear you, yeah.
Okay. Which one were we talking about again? Sorry.
So this is the group that debated the origins of life, biogenesis versus spontaneous generation. Which group of scientists showing on the screen do you think best matches that contribution?
I think that it is the Reddy, Needham, Spallanzani, and Pasteur.
Great job. Yes, that's correct. And to the nearest century, what would you say? This is actually a tricky one because it spans some time, but what would you say?
I'm not sure.
That's absolutely fine. We'll do a raise your hand for that if somebody wants to say what they think. Go ahead, Haley.
Well, it's kind of spread out, but it seems like mostly in the 1700s is when they were doing it.
Yeah, good job. So it's roughly from 1660 to 1860. So it does span that period of time. So good job, you guys. Thank you. All right, let's move on to the next one. Proposed and tested the presence of endospores. So think about that for a second and I'll cold call someone. Think about what you would answer if I called on you. Which group did this and roughly when did they do it? All right, let's go to Ashley. Ashley, what would you say?
I'm not sure, so I'm gonna pass.
Absolutely fine, and thank you for saying that. All right, let's go to Kiara. What do you think, Kiara?
Is it Tyndall and Cone?
Absolutely right. And do you know roughly when they did that?
Sorry, I don't.
That's absolutely fine. Let's do a raise your hand for that one. Who would like to say what they think is the rough period of time when that was being done? Go ahead, Madeline.
In the 1800s, like the mid 1800s.
Great job, you guys. All right, next one. Investigated the germ theory of disease. Think about this for a minute. Which of the remaining groups investigated the germ theory of disease, not as a group, but individually? They can make contributions to this idea. Think about which of these options that are remaining you would choose if I asked you, and then roughly when that was happening. Okay, I'm going to go to Todd. Todd, what would you say?
Is it Bassi? I'm not going to be able to pronounce those names. Lister and Koch?
Yes, absolutely right. Bassi, Semmelweis, Pasteur, Lister, and Koch, or Koch is the two ways that that's usually pronounced. Do you happen to have an idea for when?
Like the late 1800s?
Great job. All right. So then we've got this. This should be an easy one, so I'm not going to give you too much time to think about it. First to observe cells, Sophie, what would you say?
Is it the Hook one?
Yes, it's Hook and Leeuwenhoek. Very good. And do you know when?
Was it like 1700s or 1600s?
1600s is right. Good job.
Okay.
Okay, so then we have Jenner, Ehrlich, and Fleming. I'd like you guys to think about what you know that they did and come up with a summary that would sort of, nicely sort of summarize what their contributions have in common. Okay, think about what their contributions have in common. And because this is a little more challenging, I'm actually going to do a raise your hand for this. So when you have an answer for what you think they have in common, how would you sort of high level summary? Sandra, or Sabrina, I'm sorry, Sabrina, I saw your hand go up.
They all had to do with treatments like vaccines or penicillin or trying to discover a magic bullet.
Yeah, excellent answer. Developed treatments for disease is what I have, but your answer is perfect. Okay, now who would like to guess, or not guess, say what sort of period of time this was? I'll give Sabrina first shot.
Sorry, I've muted myself already. I assume it was a little bit later than them because they seem to be organized in like date order, so late 1800s.
Yeah, so it was actually the 1790 through the 1930s. So this is a pretty wide range, right? Jenner was first and then Ehrlich and Fleming came later. All right. Good work, guys. Okay, so this is going to be something I'm going to give you a prompt to think about, and then you'll raise your hand when you're ready to answer. Okay, and so I'm just going to, I don't know if I can do this, huh? Lower all hands. Okay, there we go. If you have a question right now and I accidentally lowered your hand to clear the boards, just go ahead and raise it again so I know that you have a question. It's just not left over. Okay, so let's suppose that you're repeating Pastor's swan-necked experiment. This is something we talked about in the chapter 1 video lectures that you would have already watched. Suppose you're repeating those experiments that he did, but you keep getting a different result than he did. So he got this result where as long as he bent the swan neck, even though it was open to the air, as long as he bent it so that the air was flowing in like horizontally, then microbes would not grow. But when he broke the neck or when he kept it straight open to the air and things could come down directly from the air, then things would grow. But let's suppose you're repeating this and you keep getting the result that everything, whether the swan neck is there or not, everything is growing. So why do you think this is happening? Let's go to Seth. What would you say?
Could it be the material? Oh, can you hear me?
I can hear you.
All right, sweet. Could it be like the kind of material you were using for the swan neck?
For the swan neck. So
Yeah.
say more about that.
Oh, is it still like, I think, at least from what I remember, at least it's I think throughout time they use different materials. So I think one they use like hay or something like that. They use like using different materials.
So I'll just sort of rephrase what you said, but you're absolutely on the right track. So the material used for the broth that was put into the flask was sometimes hay and sometimes other things. So that was when people at the time were looking over Pastor's data and other people's data and trying to figure out why some people weren't getting his results replicated. They did notice that it came down to which material was being used to make the microbial broth. Let's go to Tiana. Do you have something to add to that? If it's a difference in the broth, what more specifically could we say?
I was going to say the same thing, but you could also look at if you're not like disinfecting or using like clean material that could already have microbes in it. So if you're skipping that step, that could affect your results negatively.
Yes, absolutely. So if you did his experiment wrong, basically if you didn't do the sterilization step, you could get this result. Let's assume that you did it right, and yet you're still getting a different result. Let's go to Jade. What would you like to add?
So I was thinking that when you heat it, some things can die or not be there anymore after you heat it at different times. For some, it might take 5 minutes, others 5 hours. So it's how long you heat it that can vary.
Okay, so some organisms tolerate boiling temperatures differently than others. So for instance, although this didn't turn out to be the main reason at the time that Pasteur's results weren't always replicated, there are some organisms that are heat resistant naturally, like the ones that live in like hot springs and stuff. That would be harder to kill using the sterilization step. You'd have to go longer, or you'd have to use a higher temperature, or even potentially a different temperature altogether. What was it that historically turned out to be the real reason, and it is based on this idea of the materials being different? What about the materials was different? Why did it matter if it was straw or not straw? Let me just clear the boards. Go ahead, Dominic.
Sorry, I kind of lost connection for a second, but to answer your question, your first question, it could be just simply that the data was recorded incorrectly.
Okay, so there could be a sort of a human user error or just a bad science. Let's assume that it's good science. Let's say, Tiana, go ahead. Your hand went up again.
Does that have something to do with like the proteins or the glucose that's in the broth that would make it?
In this case, that wasn't the reason, although at another time that could be a good hypothesis, and I like where your head is at. Megan, why don't you let us know what you're thinking about?
I may be cheating, but I think I remember from last time I took this class. Does it have to do with endospores?
Yeah, tell me more about that.
Endospores are it's a thick coating around the nucleus that's heat resistant.
Yes, you're on the right track. It's not quite, what it is the cell creates a second copy of its DNA and it wraps it in a spore coat so that if the cell dies, the spore is preserved, and that spore is heat resistant. And because the alternative material sometimes used in these broths was straw, it was full of dirt, which contained soil organisms that are endospore forming. All right, good work, you guys. So now we're going to go to a chat exercise. And Tiana, your hand went up again. I just wanted to check and make sure that I didn't miss something you wanted to add. No. Okay, it went down again. Okay, so now in the chat, I'd like you to write the 3 domains of life. This should be straightforward review from cell biology. Good. All right, so there are your 3 domains, just like everyone is putting in chat. And which of these domains contain microbes? Which of these 3 domains of life contain microbes? Correct, it is all of them. So definitely bacteria, which are all microbial, and archaea, which are all microbial. Both of these are single-celled prokaryotic cells, so the smallest cells. And then eukarya, which contains us, also has protozoans, algae, and some fungi that are microscopic. Are there any non-living microbes? And if there are, list them in the chat. Probably there are, because otherwise this would be boring to answer, right? Don't forget, you can always crib notes off of other people in the chat, because this is a collaboration here today. This is not your exam, so you are allowed to work together. Also, I'm not grading you. Good. Yes. So as was described in the chapter 1 video lectures, there are 3 categories of non-living microbes. They're infectious agents that infect cells, and they're considered part of the microbiology sphere because they cause disease, even though they're not cellular and not technically alive in the same way that cellular life is alive. But viruses, which have the most similarity to living things, but nonetheless lack some key features of cells, viroids, and prions. So here is kind of a summary of all these different groups: the bacteria and archaea, which are our single-celled prokaryotes. And then we've got our protists, the protozoans, and the algae, which are animal-like or plant-like. And we've got fungi, right, which are that include single-celled yeast. And some fungi, which are multi-celled, are also microscopic. We've got viruses, viroids, and prions. Okay, so this is going to be more for fun. And we're just going to do it really quick to see, if we know what sort of causative agents there are for disease, and also to kind of get a sense of the variety. So as I call each disease, I'd like you to write in chat if you think it is caused by bacteria, virus, fungus, or parasite, or other, if the thing that is causing this is not listed here as one of the options. And other you can say, or you can actually say specifically what you think it is. All right, let's go starting with typhoid fever. Great, so we have a kind of split here. Some people saying bacteria, some people are saying virus, and some people are saying parasites. All right, I'm gonna go next to salmonella. All right, everybody pretty much knows that salmonella is a bacterial species or a genus and has many species, some of which are pathogenic. That's true. Mad cow disease. Oh, the power of the press. Most people are saying prion, and that's accurate actually. Malaria. What do you think malaria? What's the causative agent for malaria? Okay, so for malaria, we're getting kind of, again, a split between parasite, virus, and bacteria. So that'll be a fun one to discover. Hopefully this one's easy. Flu. no judgment if you're not sure, but hopefully you know that the flu is a virus. It's pretty famous. All right, what about rabies? Okay, most people are saying virus, and that is true. What about polio? What about polio? Okay, so here we're getting like a 60-30 split between bacteria and virus. So that'll also be a fun one to reveal. What about the common cold? Yeah, virus. And AIDS? Caused by, yes, also a virus. If you didn't know that, you've led a very sheltered life. Syphilis, syphilis. What's the causative agent of syphilis, do you think? It's a terrible STD. It's actually worse than you think it is probably, unless you've been watching unless you've been binge watching some of the things on Netflix that happen to have historical accuracy. All right, so most people are saying bacteria, and that's accurate. What about herpes? Yes, it is a virus. What about gonorrhea? Another STD. Pretty much everyone agrees it is bacteria. What about smallpox? Yes, one of the most famous viruses of all time because we managed to get rid of it. Amazing. That's a huge global effort, but we did it. Pertussis. What do you think, pertussis? All right, so the vast majority of people saying virus. That's actually incorrect, but it is the common conception. And probably most people think of it as a virus because there is a vaccine, but you can vaccinate against bacteria too. Tetanus. This one's also a mix, so that'll be fun to look at. So I'm about to reveal the answer key, okay? And just have a peekaloo and see which things that you were wrong about, you know? And also have a look at all the colors on this board. You know, it's a pretty, there's a lot of everything, isn't there? There's even fungi can cause some of these things, although I did not put those up here, but fungi are also infectious agents in some cases. All right, now we're gonna do this little exercise. Let's see what our time looks like. Oh yeah, we're doing well. So one of the things in the video lecture that's really important is you were introduced to all these groups of microbes and what their sort of major features are. Dominic, go ahead, you had your hand up.
Is fungi mad cow disease? Is that the other?
No, but good guess. No, actually it's the prions. So I don't know if you guys will remember the whole British beef scandal, and then of course it showed up elsewhere. But proteins, prions are an infectious protein that is found in diseased brain tissue. So if you eat it, then you can get a degenerative brain disease called mad cow disease in cows. It's called Creutzfeldt-Jakob's disease in humans, and it's called scrapie in sheep. So what happened was our food stream was changed so that cows started getting fed rendered sheep. And because some sheep get scrapie, that's actually pretty common. The disease protein ended up in the feed of the cows. It went to their brains. They got sick. And then when it was fed to people, they got sick. Fun experience to have one of the many side effects of modern agricultural practices. All right. Great question. Thank you for asking that. So here are your groups of microbes that you're responsible for in this course. I'd like you to take a little bit of time to write down what you can remember about their size generally. So they're going to be sort of a rough size range. Their composition, so what are they made of? You know, if it's cells, you can say that. Structures, so what are distinctive structures? You don't have to list every structure, but is there a distinctive structure they have or do not have? And whether they're in the living or non-living category. So just take a few minutes to do that, and then I will cold call you guys to find out what you remember. Sorry about that. I was muted for a second. Hopefully that was long enough to get a sense of this. And again, you can always pass if I call on you and you're not sure, but what I'd like you to do when I call on you, and I'll tell you what group you're responsible for, just saying what you remember about its rough size range, what it's made of, structures that are important or missing or present that are sort of defining. Anything that you can remember, like does it have a cell wall? And if it does, what is it made of? What is its size? Does it have organelles? Does it not have organelles, etc.? Is it living or non-living? Is it cellular or not cellular? Okay, let's begin. Where's my participations box? I lost that for a second. With Alexandria, what would you say about bacteria?
I wrote down it's about 1 micrometer. Its cell walls are made of peptidoglycan, and it's a living organism.
Excellent. Okay, good job. Let's go to Gloria for archaea. What would you say, or what did you write down?
I didn't get much, but I said it's a non-living thing, non-living organism.
Okay, great. Thank you for that. So let's add another person. Let's add Kendu in and see if you can add more to this answer. What did you write down for archaea?
I'm not sure about that.
Okay, so you want to pass?
Yeah, I want pass.
All right. Let's go to Lauren. What would you say about archaea? Are you there, Lauren? I can't hear you. All right, Dominic, I see your hand go up. Is it because you want to answer? Uh-oh.
I figured I might as well, since nobody else was. That archaea, I know that they are extremely old and that they can survive extreme temperatures, whether it's cold or hot. And you can find them still today in like hot springs and stuff like that.
Yes, so there are extremophiles in this group, although it is important to say not all of them are extremophiles, though some of them are, many of them are. And there are also extremophiles in the bacteria group, but that's right. And is there anything else you want to add to that?
Nope.
Awesome, thank you for that. Savannah, your hand went up as well.
I think one of the important things you mentioned was that they're really similar to bacteria in all ways, except for really the peptidoglycan was one of like the major distinctions.
Right, yes, they are very similar to bacteria, both in their size, which as the previous answer for the bacteria mentioned was about 1 micrometer range, and their cell wall is made of a different material. Yes. Ruby, your hand was up as well. Did you have anything you wanted to add?
Yes, I couldn't remember if the first person who spoke said it was living or non-living. I was under the understanding that it's a living.
That's correct. Yes, it is cellular, just like bacteria cellular.
Oh.
Thank you for adding that. That's great. Thank you, Ruby. Okay. Let's go to fungi, and let's see. I am going to hit up Noor for fungi. What did you say about fungi?
Did you say Noor?
I did.
Oh, okay. Fungi, they get their energy from degrading organic stuff.
Yes. Did you have any other notes, or did you want me to move on?
And it's a single celled, like
okay.
For example, yeast and like mushrooms for multi-celled.
Yes, good. Yes, so fungi contain both multi-celled and single-celled organisms, including yeast, which are single-celled. The macroscopic mushrooms are not microbes, but there are multi-celled fungi that are microscopic, absolutely. And they are heterotrophs, as you mentioned. They, like us, they have to eat organic chemicals in their environment rather than creating their own food the way that photosynthetic organisms do. Let's see, I'm just going to... who else would like to add anything about fungi that hasn't already been said? Seth, go ahead.
Was it they're also, was it they're not photosynthetic?
They're not photosynthetic, that's correct. They're not photosynthetic.
Also, actually, I have a question too. Actually,
Yeah,
so
go ahead.
so what distincts like the psychedelic mushroom versus like the regular mushrooms that we eat? Is it just from like the material that they eat compared
What
to?
distinguishes them is their genetic code, and that makes certain proteins that are enzymes that create either psychedelic compounds or not. So it has to do with the genes and which proteins and enzymes they have.
Okay, so that's why there's some mushrooms you can eat safely, and then there's some that
Yes.
just make you go psycho.
Yes.
Okay.
And it is worth mentioning and sort of reiterating that the mushrooms that we eat are not microbes. They're macroscopic. We can see them.
Oh, got it. Okay, thank you.
So we don't consider microbes or mushrooms in this section. Let's see. I see Ferdowsi, you raised your hand. What's on your mind?
I just want to add that they are eukaryotes and they have membrane bound organelles, but I have a question whether or not they have mitochondria.
Oh, great question. Yes. So the fungi are eukaryotic, as Ferdowsi mentioned, and they are they do have membrane-bound organelles, which is a defining feature of the eukaryotic group. And they do have organelles. We'll talk more next week, not next week, actually the week after, about what specific organelles are found in these different eukaryotic cells. So that's a great question that we'll come back to. And I think that was all the things that you added. So Ferdowsi, thank you for that. Does anyone want to raise your hand for a size range on what these fungal cells, what's their size range? Go ahead, Cindy.
Aren't they like 400 around there?
So they can be quite large. Some of them are smaller. The single-celled yeast do range in the sort of smaller 10 micrometers, but there are fungi that get much larger because they're very long. So yes, they are in that size range of eukaryotic cells. We typically say that's 10 to 100, but it can be larger, as you mentioned.
Okay.
Good job. Okay, let's move on to the protozoans, and I'm going to tap, let's see. Oh, Hannah, you raised your hand. Did you have a question or wanted to add something?
Oh, no, sorry, it was an accident.
All right, thank you. Shutong, we're going to go to protozoans. What did you write down about them?
Can I pass?
You absolutely can pass. Yes. Let's go to Mia. What did you have to say about protozoans?
For protozoans, I said they're single-celled but complex. Most ingest organic compounds. They have no cell wall, and most are mobile via flagella.
Yes. So they're single-celled, they're eukaryotic, and the protozoas, because they're animal-like, they're typically heterotrophs, just like we are and like fungi are. And they don't have a cell wall, but they do have flagella. And the fungi, I forgot to mention, have a cell wall. And does anyone know what it's made of? The fungal cell wall. Raise your hand if you know what it's made of. Madeline, go ahead.
It's chitin.
It's chitin, that's right. Good. Let's go to algae. And let's hit Rachel. Rachel, what did you say about algae?
So for algae, I said that it's single or multi-celled, that it's photosynthetic, and the cell wall has cellulose.
That's right. And is it a eukaryote or a prokaryote?
It's a eukaryote.
That's right. Yes. So it's going to be in that, each individual cell is going to be in that larger size range. Good job. Let's go to viruses and let's ask Teresa. What did you write down about viruses?
They are acellular. They will infect a host cell. Their core is DNA or RNA. And some are enclosed by a lipid envelope.
Excellent. Yes, viruses are acellular, meaning they don't have a cell. What they have is genetic material that's either DNA or RNA. They're enclosed by a protein coat and sometimes a lipid envelope. Good work. All right, viroids. Hannah Hubble, what did you say?
They're simpler than viruses. They require host for replication, single short RNA, and no protein coat.
Yes, they're made of a single, very small RNA. It's tiny, actually, smaller even than a gene, typically. And they are, like viruses, acellular. They replicate inside of a host. And prions, last, let's go to Elizabeth Prions, what would you say about them? Elizabeth, if you can hear me and unmute.
Oh, sorry. They're non-living microbes.
Yes. And what are they made of?
Proteins and stuff.
Yes, they're made of one protein, an infectious protein. Good. All right, so here's kind of a very quick summary of what the very most basic information that I would expect you to remember for an exam. So you can use that to review. I'm going to skip this for now unless we have time for it later. And we'll do this, I think. Let me look at my time here. I kind of want to do this right now. So I'm just going to give you a minute to think about what you would say. Write down an answer to this prompt. How do microbes affect humans? Okay, and as soon as you have an answer, raise your hand if you'd like to share. And I'm just going to do okay, go ahead, Dominic.
Sorry. Microbes are important for the digestive system.
Yes, that's absolutely true. Let's go to Megan.
Obvious answer is that most of them can be pathogenic to us, and they can also affect our food source, such as plants.
Yeah, they can make us sick, and they can also destroy our crops. Very good. Jade, go ahead. Your hand went up as well.
That we could not survive without them. They do things that we can't, like the nitrogen fixation, carbon fixation, and the decomposition of organic materials.
Yes, in the ecology of our world, they're absolutely critically important for recycling nutrients back into a form that we can actually use and access, degrading things and building things. Absolutely. Manuel, what would you say?
Hey, well, I want to add that maybe, microbes, like humans have like domesticated some microbes to use them in industry, especially for food, like beer.
Yes. Yes, absolutely. Yes, they do metabolic processes that are very handy for us. So they have been used in a variety of food and production and industrial processes. And Savannah, what would you like to add?
That they're easily studied cells relative to like studying a pig or studying a person, so we can learn from them.
Yeah, absolutely right. They are good model systems for studying and research and understanding some of the shared things that we have in common with them. Sabrina, what would you like to add?
It's also, hold on. Okay, it's also important to understand them so we can develop aseptic techniques for handling them, for surgeries, and in research.
Yes, absolutely true. Ashley, what would you like to add?
That they also contribute to non-infectious diseases.
Yes, they can. They can. Yeah, yeah. There are a few examples of that I can think of off the top of my head, such as tetanus, which can cause a disease which is not really infectious between hosts, right? You get it by, having a deep puncture wound typically, but it is a microbial disease, and you can catch it from the environment, but you can't pass it to other people. Good answer. And for Firdausi, what would you like to add, or what question do you have? I don't know if you can hear me, Firdausi. You're muted, so I can't hear you.
Oh, sorry. I was gonna say that they help us digest and ferment food in our system.
Yes, exactly right. Yes, you and Dominic both mentioned this very important thing, which is that they are part of our digestive system. They're also part of our, they cover our whole bodies, and they're important in lots of body parts, not just the GI tract. Good answers. Great job. All right, you guys. So there's a few slides in here that kind of touch on things that microbes do that you can just look at when you're reviewing this. And in the video lecture, I discuss sort of the situation currently, the fact that we don't know everything, because it turns out the more we find out about the world, the more we realize that there's many, many species that we have never ever studied because we can't figure out how to cultivate them or didn't even know they existed. So it's a whole new amazing world out there of discoveries waiting to happen. And the other thing that was discussed in the chapter 1 video lectures is the importance historically of microbes and disease, and also our current challenges, which include things like antibiotic resistance for bacterial infections and evolution of viruses. And of course, as we well know, our current situation, which is an inevitable result of the rapid pace of viral evolution and species transfer. So what we're going to do now is take our little 10-minute break. We're going to come back at 5 minutes after 11 and talk about lab stuff. So I will see you shortly. Welcome back, now that I'm not muted. Let's just have a quick yes in the chat if you can hear me. Fabulous. Okay, so we're going to dedicate the next part of this to lab. This is a lot more time than we'll usually spend talking about lab. But I kept the first, this chapter 1 sort of session short and sweet so that we would have time to discuss it. The first thing I want to do is a very quick breakout, just to test my breakouts, to make sure that I have put you in the correct groups. Okay, so I used the information you gave me at the beginning, plus my own notes, and I did see a note from Sabrina earlier, which I now have lost in this giant text session, that you were not in the correct lab group. So I'm going to just, Sabrina, if you can hear me, just unmute real quick and let me know what lab group you should be in.
It doesn't really matter, but I was signed up for lab group D and I'm no longer signed up in any lab groups.
Huh. Okay. Well, that one is full, but you are in, according to my, oh, but I signed people this morning. So let me have a peek, and you're going to end up in the group that I put you in, probably is what happened. Did you, well, we can talk about this more. Stay behind after class if it matters to you which lab group you're in, and we'll see what we can do. But where are you? I put you in one of these and now I can't find you. Hold on 1 second. Sorry for the delay. That will allow anyone who is straggling to join us. I put you in K because you weren't in a group this morning. So that group, okay, so I'm going to send you off to your lab groups, and then if there's any issues with the lab groups, we'll just, we'll check back in a minute, and also you can stay at the end of class for a more serious issue. But I'm going to send you off to the groups that you are currently in according to my notes. And just for 1 minute, just so you can all say hi to each other and make sure that you're in the correct group. The lab group that I'm sending you to, the room has a name, the letter, and so that should be a clue as to whether you're in the right place. So you should see an invitation. Go ahead and join your groups. Okay. Awesome. So the breakout rooms are closing in 30 seconds. As you're returning to the room, don't forget to turn your video off and make sure you're muted. You should be automatically muted, but you want to make sure that you don't have anything being broadcast that you don't want to. That's happened to all of us, hasn't it, in Zoom this year? And then let me know if you experienced any, if you were in the wrong group, or if anything was going on that was concerning to you. You can private message me if it was very egregious. I don't expect people had enough time to be super mean to each other just yet. All right, I see your message, Sabrina, thank you. Okay, welcome back everyone. Hopefully that was successful. Did anybody have trouble getting into their breakout and just didn't get there? Yes, Ruby, you only had 2 people in there. Let's see if that's because of, that's expected. Yeah, Ruby, there were only 2 of your members here today. That's why.
Okay, I thought we lost connection, but when there was only 3 of us, that was our group. Is that right?
Yeah, so some groups only have 3 and others, there are some people missing today.
Okay, thank you.
Yeah. Any other concerns? All right, we're gonna do this again later. We're gonna meet for a bit longer so you can actually talk to each other about lab and not just stare at each other's faces awkwardly. Let's talk about the lab logistics, a reminder of how this works. So each week that a lab is assigned, the prep is due by the Sunday of that week. So for instance, we're in week 1, this is lab 1. You need to have read the lab 1 background and lab 1 introduction, instructions. That was a typo on my part. I will fix this. Lab 1 background page and lab 1 instructions page. Use that to answer the pre-lab quiz. All of this stuff is due by Sunday night. Also due by Sunday night is contacting your lab mates and getting organized, posting at least twice in the lab 1 discussion. So if you're paying attention, then what you notice is you better read and do all this stuff well before Sunday if you're going to get to a chance to productively coordinate with your team by Sunday night in the discussion. So I'm grading this pre-lab quiz, and I'm also grading the discussion early in the quarter. Later on, I'm sort of leading you to your own devices, but right now I'm going to make sure that you have posted at least twice in a substantive way that indicates that you're interacting with each other and making progress. And now those posts are due by Sunday. Every lab has its own discussion just so that you don't get kind of a massive tangled web of things. So for lab 2, you're going to have a discussion you need to post in that is about lab 2. So then next week, you'll basically have all that time to do the lab itself. You can, of course, get started right away. I have some people who are already messaging each other about the content, the deliverables for the lab that they've already finished. Not to stress you out, but just to motivate you to do stuff. You know, don't wait just because you can. Get stuff done as soon as you can. That's really the best plan. But you have until next Sunday. So this Sunday, pre-lab stuff is due, pre-lab and discussion. Next Sunday, the actual lab materials that do things like the tables that you have to create and the conclusion that you have to write, that's due next Sunday. A reminder that the tables that you create that are collaborative can be the same as others in your group. If it's an independent one, I expect it to be independent. So there's one in lab 1 where you have to write descriptions on your own. And so those should be your own descriptions. I don't want to see everyone's being the same. But for the collaborative tables where you do research together and share data with each other, then it's fine if it's the same identical table from everybody in the group. But nonetheless, you have to each submit it. I'm not going to go hunting through my grade book to find the one person who submitted it for their group. And don't forget to write a conclusion. That's described online, so you'll do that. There's also a post-lab quiz that's due at the same time as the tables and conclusion. So let's talk about what is important in lab 1. Basically, it kind of helps us to look at where are microbes found and how do we count them. So the microbes found would just take 1 second. We're not going to do it for 4 minutes. I'd like you to think about any one of these prompts. Just write something in the chat about any one place, either in the natural world or in your home or your workplace or in or on your body, one place where you would expect to find microbes. Write that into the chat right now and I'll just read them out as I see them. Good question, Seth. Remind me to answer that a little bit later about the lab manual. Okay, so let's read these out. So skin, mouth, hands, kitchen sink, intestines, dog, kitchen counter, couch, phone, armpit, face, bathroom, surface, nose, GI tract, eyebrows, keyboard for sure, puddle, door handle, any mucous membranes, food, yep, toilet for sure, ears, outer space, Wow, I love that one. It depends where you are in outer space, and we don't know a whole lot about it, but there's, yeah, actually, I think I would have to look that up a little bit more and sort of figure out which things are where. Obviously, we haven't explored all of outer space. Doorknobs, pencils, tap water, yes, soil, gardens, and under your fingernails. Okay, good job, great. So I think that sort of helps to illustrate that microbes are many places. It's not actually fair to say that they're everywhere normally. There are parts of your body that do exclude microbes if they're doing their job. And if you find microbes there, that's the sign of a person who is in trouble. Okay, now let's ask this question. Which parts of the human body are normally colonized in a healthy state when you're not diseased? So I'm just gonna do, we're gonna do yes, no's for this. And for this, we'll go to our participants box or the reactions, wherever you find the yes, no option, you know, the little green check mark and the little red X. So as I call them out, you're gonna vote, okay? Skin. Yeah, most people said yes. Ear canal, external part, and nose and throat. Firdausi, what's your question? Oh, was it?
I apologize.
Yeah, it was an accidental raise your hand. I get it. I know it's in the same place, isn't it? Stomach, small intestine, and colon. Okay, yes. External genitalia, vagina, and urethra. What do you think? Okay. What about the bloodstream? Okay. Most people are saying no, which is accurate. Internal organs such as heart, kidneys, and spleen. Okay, a lot of no's, good. And what about your subcutaneous fascia? Yeah, normally you wouldn't want it there either. Okay, so the answers are not shown, but just to sort of like everything above here, these first four categories, you would expect to find normal microbial colonization. And down here, internally in the body, you would not. All right. Let's look at our time. I might skip over this little, oh, let's do it anyway. So let's imagine, okay, we're just gonna imagine we do an experiment. We're not doing it, we're just imagining that we swab an area with a cotton swab, let's say, and we cover the surface. We use that cotton swab to inoculate a rich medium bacterial plate, and we incubate it for a couple of days. This is gonna grow bacteria and yeast or fungi primarily. One cell from that little swab will grow into a colony. So my question for you is, how many cells are in that colony? Okay, let's start with that. So raise your hand if you have an answer to that you'd like to say. Or if you want to put it into chat, that's also fine. Billion, says Dominic. Anyone else want to? Oh, good question, Kayla. I'll answer that in a little bit. Firdausi says too many. Who else wants to hazard a guess? I'll wait for one more answer. Okay. So yes, the answer is usually billions. And Sabrina says, are you able to count it? Yes and no. You can't count it just by looking at it, but you could take that colony, suspend it in liquid, and then do cell counts. There are ways to do that. Not all microbes can be sampled this way. That is to say, not all microbes, if you swab an area and run it over a rich medium, will actually grow. And what is the reason for that? Let's do a raise your hand for that. What is the reason that you cannot sample all microbes in this way? Sabrina, go ahead.
If I remember right, not all microbes are able to survive in lab environments.
Yes, that's right. And I mean, it's sort of a roundabout question, because really what that means is not everything we know what conditions are required and have been able to sort of generate that required condition in a lab. Manuel, what would you like to add? Not all microbes can be sampled this way, and why? Can you hear me? I can't hear you, Manuel. You're muted.
Sorry. So yeah, no, not all microbes like survive in oxygen. You know, with an oxygen source.
Yes,
And
exactly.
it depends like what the, the, the plate has.
Yes, exactly. It depends what the plate has. It depends on the gaseous environment. And these are all things that we need to know and may or may not be appropriate to the particular experimental technique that we're using at that time. Frankie, did you want to add anything?
Yeah, I was just going to say that some microbes need a host to reproduce.
Yes, like viruses, for instance. Good job. Yes. And even so-called free living like bacteria may require a very complex ecosystem that can only be provided by, by cohabitating with other organisms usually. So we might not know how to do that. Okay, so for this lab, you want to do the background and instruction pages reading and then use that to answer the pre-lab quiz. And for the lab 1 activities, what you're going to do is collaborate on researching species, different species and where they might be found. And then independently, you're going to do these calculations. And this is a measuring of how much stuff, how many microbes are in an air or water sample. So I want to walk you through that process of the math, and we can revisit this next week if you need to. But we're going to start with air and water, and then I'll actually walk you through some visual examples. So just to first talk our way through it, and then we'll sort of look at it in a more visual way. If we want to sample an air sample, one of the standard metrics is something called CFUs per square foot hour. What that means is if you had a square foot, an area, a surface like the surface of your desk or floor that was a square foot in size, and you waited a whole hour and counted how many things landed on it that could grow, that would be your answer. So that kind of gives you a sense of how much stuff is in the air if you count how much stuff lands in a square foot every hour. So to do this kind of test, you take a plate, solid agar medium containing food, open it to the air, and you expose it for a period of time. Usually the plate is not a square foot, and usually the exposure time is not an hour. So you have to then convert the answer that you get. So you take your plate, you expose it to the air for however long, and then you close it and you incubate it for 2 days, or whatever period of time you deem is appropriate. And then you go back to it and you count how many colonies have formed, how many things fell on that plate that could form colonies. Obviously, this is actually not a measure of how much microbe is in the air because there could be viruses that won't grow. There could be bacteria that don't use that particular condition. This is a measure of CFUs, colony forming units, that can grow in these conditions. For Dowsey, go ahead. What's your question?
So do you do that without opening it? Do you count it without opening it after incubating it in?
Yes. So to do the experiment, you have to open it, obviously, so things can fall
Yes.
onto the plate. But then you close it once the time period is up. You close it, incubate it, and then you keep it closed.
Okay.
Yeah, great.
While counting, right?
Yes.
Okay. Thank you.
Yeah. It doesn't really matter if you were to open it for like 5 minutes while you counted. That wouldn't be enough time for newly dropped cells to grow colonies and mess up your math. But we keep them closed because we usually don't know what's on there, and it could be not so good for you. Okay, so we have to take that plate and we've got to convert it, because we didn't use a square foot sized plate. We used probably something smaller, and quite common. It's a 10 centimeter plate that's 1/15 of a square foot. And we usually don't use a whole hour because we're impatient. So we might use like 15 minutes, which is what fraction of an hour? Put that in the chat. What's 15 minutes fraction of an hour? A quarter, that's right. Okay, so if you had done it this way, and you could do it lots of ways, so this is not the only way, but if you had taken this size of plate, which is this area, 1/15 of a square foot, and you had exposed it for a quarter of an hour, and the answer on the plate was there were 10 colonies, what do I have to do to that number in order to actually get my answer, which is how much would have fallen in a whole square foot, not a 15th, for a whole hour instead of a quarter of an hour? What do I have to do? Raise your hand. Okay, I'm seeing in the chat too. We have to, yeah, Menhui says we have to multiply by 15 and 4, right? So we have to multiply by 4 because we only exposed for a quarter of an hour. So whatever, if we get 10 on this plate in a quarter of an hour, we would have gotten 10 times 4 in a full hour. So we take our 10, multiply by 4, and we get 40. But we also only measured a small area, a 15th of a square foot. So what do I have to multiply this 40 colonies by in order to get my answer, which is per square foot. I've already multiplied to account for the time, and I have to multiply now to account for the area. So I multiply by 15. That's right, Savannah. Okay, so we will walk through this again, if that didn't quite make sense. And this is on the recording, so you can revisit it. Let's talk about water samples though. So in a water sample, the area is irrelevant. The volume is actually irrelevant, not the volume. The area and time is irrelevant. What we want to know is in a particular water sample, how many colony forming units, so how many microbes that can actually form colonies, are there per milliliter per volume. Okay, so if we have a water sample, let's say from your toilet bowl, or let's say your tap water, or let's say a pond in your backyard, and you take a known volume, a milliliter, or usually it's going to be 0.1 of a milliliter, so like a tenth, and you spread that on a plate, and then you incubate it, the size of the plate is irrelevant. What we want to know is in that volume that we've already taken, how much stuff is there? So we just count the colonies directly. Quite often the original water sample is too full of microbes for us to get an answer that would be useful, so we use a dilution. All right, so let's suppose that we were sampling your tap water and we took this tap water and we did a 1 in 100 dilution. Okay, so we took 1 mil, let's say, put it in 99 mils, and so now it's 100-fold diluted. Whatever the original concentration was, it's 100-fold less now. And we take a tenth of a milliliter of that and put it onto a plate and grow it up, and we get 10 colonies. What do we have to do to get our answer? We have 10 colonies per tenth of a mil of a 1 in 100 dilution. So what do we have to multiply this by? Yes, so someone wrote, we have to multiply by 10, right? We have to multiply by 10 because we want to know how much is in a milliliter, but we only sampled a tenth of a milliliter. And we have to multiply by 100 because we diluted it 100 times. Okay, so now let's look at the actual way to do this. We'll do a little walkthrough here. Okay, so here is a hypothetical air sample plate. So it's a Petri dish, it's a 10 centimeter plate, and it was left exposed to the air for 30 minutes, half an hour. Okay, so let's first think about what the area is like. So this is a 10 centimeter plate. This plate is 1/15 of a square foot. If we wanted to not have to do any math, how many of these plates would we have to leave out and count in order to get a full square foot? Put that into chat. 15, that's right. So instead of doing that, which is wasteful, we can just take the number that we get on this plate and multiply by 15 to say, if we had 15 of these plates, which would cover a whole square foot, we would just take this number and multiply it by 15. Now we have exposed for a half an hour. We really want to know what would have happened in a whole hour. So what do we have to do to our number as well to account for the difference in the exposure time? That's right, we have to multiply it by 2. Okay, so we could expose this for a whole hour, or we could expose it for less time and just multiply to make up the difference. All right, so the first thing we have to do with a calculation like this is count colonies. You'll do this in lab, and in a test, you could be asked to do this calculation, but I would give you the CFU number so you don't have to waste your time counting a plate. But let's do it. Okay, so we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 5. Okay, Dominic, you had a question.
Stop you because you're on a roll too much and it's kind of off topic, but I just wanted to check on the status of the lab material and see if you heard anything when you're done with this.
Yes, so let's revisit that at the end. Great question, excellent. And Savannah, you also had a question.
For examples like this, are we always looking at colonies that'll look the same, or are we just colonies in general? Or will
colonies, yeah, so it's a great question. These are not the same. There's subtle color differences which do indicate they're different species. This is a rather uniform sample, but there could be really big colonies, like a big honking colony over here, and really tiny speck-like ones, and they could be all different colors. So we're counting the actual number of, like, whatever they look like, we count every single one of them. If we're counting types, then we would go, it looks like a lot of these are similar size and white, so that's one type. And some of these are kind of orangey, so that's a second type. So maybe there's 2 types on this plate. But in this calculation, we count every single colony. Does that help?
Yes, thank you.
Awesome. Okay, great question. Okay, so this is our CFUs on this plate. This samples a 15th of a square foot. So what do we have to do to this number to make the, to account for the fact that we didn't sample a whole square foot? Oh, I'll get to that question in a second, Sabrina, good question. So what do we do to this number to account for the fact that we did not sample a whole square foot? We sampled a 15th of a square foot. Multiply by 15, that's right. Our exposure time was 30 minutes. So what do we have to multiply this number by in order to account for the fact that we didn't sample a whole hour? We have to multiply by 2. That's right. Okay, so we would take, so our time is 30 minutes over 60 minutes, which is a half an hour. Seth, go ahead. What's your question?
Wait, so would you multiply by 25 for, like, would you multiply 25 times 15 and then 25 times 2? Is that what you would do?
You would take 25 times 15 times 2.
Okay, okay.
Yeah, you don't have to put the 25 in there twice. Yeah.
Wait, so with the CFU, is that given or do we have to figure that out ourselves?
On a test, it would be given. In the lab activity you'll do this week, you have to count it yourself, and you don't have to be 100% accurate. It just has to be close.
Okay, all right, thank you.
So this gets to Sabrina's question, which is, she said, I can see other little specks here that you're not counting. So there might be a difference in, you know, there's a little tiny speck over here and I'm not counting it. But if you did on your lab, it would only change this maybe by a couple of, you know, maybe you'd come up with 27 instead. And then that would be fine. If you're coming up with 50 instead of 25, we should talk. Also, my eyes are not great. So maybe there's things here, like there's a little one over here that I didn't count. And it would be fine for you to count those. But that's another valuable thing about, you can actually talk to other people in your lab about what numbers they got. Don't copy them, but you can sort of calibrate with them and say, hey, what did you get for this? Like, how did you do it? I'm having trouble or whatever. Okay, so what do? We can
I have a
do
question,
yeah,
please.
go ahead. Oh, sorry, Firdausi, I did not see
It's
your
all
hand.
right.
Go ahead.
So what if they match together, like they come in form of 2 or 3 numbers? Are you going to count each one as individual, or you?
So are you talking about like here where 19 and 20 are really close together?
Yes.
Yeah.
Yes.
As long as you can tell that they were probably separate, like formed by separate cells, even if they're touching, you would count them separately.
Okay, thank you.
Yeah, good question. If you can't tell, you're just going to have to take your best guess. So like I said, the lab one where you count for yourself and you come up with a CFU number yourself, you're going to probably differ from somebody else, but hopefully the difference isn't going to be like twofold difference, right? It's going to be, you know, an extra 10% or something, or a reduction. Okay, so how we would do this calculation is we take the CFUs that we've counted, or on a test, I would just tell you what that is. And we divide by the area and the time. Okay, so we can take 25 divided by a 15th of a square foot times a half an hour. This is one way to do the calculation. So 25 divided by 1/30th of a square foot hour. And that gives us this answer, because remember, when you divide by a fraction, you take the bottom and put it on top. And this is 25 by 30. Or if this does not make sense to you, this is the way to do it where you're going to end up with the right units and not ever be confused. But if you're not an equation person and you're more of an intuitive math person, you can just do this. 25 CFUs times 15 plates would be required per square foot, times 2 exposure times would be required per hour, and we get the same exact answer. So let me just take a second and ask you if you understood that, or if you'd like me to go over it again. Awesome. Okay. And if you did not, or you're feeling a little shaky, you can also stay after class and I'm happy to talk through it again. We're going to do a little bit of, and so here's the answer, 25 by 30 is 750. And this is the unit you have to use, CFUs per square foot hour. All right. Now, let's do this. So we're going to use the exact same sample, but we're going to say that the conditions were a little bit different. Okay. And we're going to see if we can come to the right answer. So this is a test to see if you did understand what we just talked about. Let's suppose we got this data, but actually this plate was not a 10 centimeter plate. Let's suppose this plate was a 15 centimeter plate, and you don't have to memorize this. I'm just telling you this because that's what it is. This is the important part that I would always tell you. The surface area of this plate in this pretend example is 1/5 of a square foot. So do we still multiply our 25 by 15? That's right, we don't multiply by 15, now we multiply by 5 because it would have taken 5 of these plates to record, to capture that whole square foot. And in this example, I'm saying that our exposure time was 15 minutes instead of 30 minutes. So what fraction of an hour is that? That's 1/4 of an hour. And so we would multiply by 2 or by something else. We multiply by 4. Okay. Because 15 minutes divided by 60 minutes in an hour is 1/4 of an hour. Okay. So here is one way to do the calculation. Again, you take your CFUs, you divide by the area, which is 1/5 of a square foot times the fraction of time, 1/4 of an hour. All of this is on the denominator. And that gives you 1/20th. So 25 divided by 1/20th, which is 25 by 20. Or you can just do this, 25 CFUs times 5 plates per square foot times 4 exposure times per hour. And that gives you the same answer, which is 500 CFUs per square foot hour. So again, this is recorded, it'll be posted and you can review it. But if at any point you're feeling a little muddy on it, please don't hesitate to ask for a little help to review it. All right, let's go to water samples. Let's check our time. I want to make sure I leave enough time for you guys to chat. All right, water samples I've counted for you. You can see that there are 17 CFUs. The volume of the water sample put onto this plate was 0.1 mil, and the dilution was 1 in 1,000. So the way to do this calculation is take your CFUs divided by the volume times the dilution. Right? The volume is 0.1 mils and the dilution is 0.001. And that gives you 0.0001. Or you can do this intuitive way. These are the same thing, basically. 17 CFUs times, it would have taken 10 plates to get to a mil because this was taken from 1/10 of a milliliter. And it would have taken 1,000 because it was 1,000-fold diluted. And this is your answer. So I'll just take a minute to see if that made sense. Checking the chats. Okay. Let's see if we can try it. So we're going to take the same data, but we're going to say that actually the dilution was different. Same volume, 0.1 mils, same CFUs, but if the dilution used to get this data was 1 in 1,000,000, then what is my original CFUs per mil? Write that into chat if you can figure it out. I'll give you a minute to think about it, because that's all we have before I have to send you off to breakouts. So do the math, see if you can figure it out. What's the CFUs per mil for this water sample? If this is the amount of stuff that grew on this plate, this was the volume used for plating, and this was dilution. So you take this, and you're going to, good, I'm seeing some correct answers out there. So if you're feeling unsure, you can have a peek at what other people got and see if you can reverse engineer your answer. You can also stay behind. I'm happy to go through this with you again. Yes. Now, just one thing, and this is fine because it's just a chat, but if it was a test, you always have to use units. So don't forget that part. You don't want to lose points over something silly like having no units, because if you don't provide units to a numerical answer, I will always assume that you're saying kittens, and sometimes kittens is the wrong unit. Okay, good. So how I would do it is take 17 divided by the volume times the dilution. The other way you can do it is 17 times 10 of these would be required to get a whole mil times 1,000,000, which was the dilution factor. And we either way get to 170 million CFUs per mil. Okay, now I don't have time for the Kahoot, so we're going to do that next week. So yay, we're going to do a CFU practice Kahoot quiz next week. And so you can prepare yourself for that. What I want to do right now is give us a little bit of time to chat with each other in a bit of a longer format. And then I do have things to tell you afterwards. So I'm going to give you 5 minutes to talk to each other. You've already met. So now is the time to talk about, if you want to exchange any other ways of talking to each other besides the discussion forum on Canvas, if you want to exchange phone numbers or emails. If you don't, that's also fine. And other people should honor your privacy. The discussion forum is perfectly fine. and kind of where you're at in this lab process. Like, I haven't read the stuff yet, but I'm getting to it, or I did read it and this is sort of what I'm thinking, or we already got these answers and now we're just going to chat about pizza. Okay, so I am going to send you off to your breakouts as soon as I find my breakout. All right. Too many control panels for me. Okay, there we go. All right, so the breakouts are ending in 14 seconds, so we'll just wait a second for the last people to rejoin us, and then we'll end the class with just a couple of comments. And there we go. All right, everyone should be back. So thanks for participating in that. When it is possible, I will give you guys a second to organize in lab groups during the week, just for a few minutes. And sometimes it won't be possible. So hopefully early enough in the term, you'll get organized and be able to be sort of self-sufficient that way. Kayla, what is on your mind?
So I'm still a little confused on what we would use the group chat for in Canvas.
Yeah, so for the collaborative parts, you need to be talking to each other about which part you're going to do, because some of the activities are ones where there's a bigger project and you kind of each take point on different parts of it. So you need to claim which parts you're going to do and make sure everyone's in agreement. And then you're going to also have to share data with each other. So that's a place to post that. Now, if you have other ways of contacting your lab mates, that's fine. I encourage you to do that. But the discussion is mandated because I need to make sure that I am seeing that you are talking to your lab people and getting organized, because sometimes it can sort of fall to the lower priority unless I make it a requirement. Does that make sense?
Yes, thank you.
Awesome. So for the first 3 weeks, it will be mandatory for you to post in that discussion. After that, you're kind of on your own, but by then I hope I've set some good habits for you, or you've decided how else you want to communicate with each other. And I do reserve the right to reorganize groups if necessary, and to kind of take you to task if you're not pulling your weight. So do kind of step up there. All right, so just a reminder of what's due, and then I'll tell you one last thing before we go. So by Sunday, you need to have read the Lab 1 background and instructions pages so you can do the pre-lab quiz. Do it well before then if possible. You need to have posted twice in the Lab 1 discussion and gotten organized so you know kind of what you guys are all doing. And then that also gives you a chance, if you're in the process of this prep and you don't understand what you're doing, to contact me and get clarification or talk to each other. By next Tuesday, you have to have read all the materials on the microscopy topic page, and you want to do the pre-quiz. This is the one that's not for credit, but this will help you prepare your mind. You want to read chapter 3 in the textbook, and you want to definitely watch the video lectures and read the slides. So then Tuesday, we'll actually talk about this material. If you're going to miss a class and you want to use your makeup, email me, or if you have other concerns. And the last thing that I want to say is, so in addition to the signing in, which you're going to do every day, you're now going to also sign out. So as you leave, you're going to write your last name, first name, and then sign out that way. You just have to put your name in it, and then you can leave when you're ready. So if you are done and you don't have questions, you can go ahead and just leave. Thank you for coming today. I appreciate you. If you do have questions or I've asked you to stay, then please stay behind for just a little bit so I can get your questions answered and address other things. Awesome. Okay. So have a great day, you guys, and let me just end this and then I'll start taking your questions. So hold tight while I stop sharing and stop my recording.